The next instrument is gel electrophoresis. That is polyacrylamide electrophoresis of proteins, and this is the structure of gel electrophoresis. Definition of gel electrophoresis. Gel electrophoresis is a laboratory method used to separate mixtures and sometimes purify macromolecules, especially proteins and nucleic acid that differ in size, charge, or conformation. And principle: changed particles under the influence of a liquid media placed in electric field will migrate to the electrode and of the opposite charge. Positive ion cation will migrate to the cathode, the negative electrode, and negative anions will migrate to the anode, the positive electrode. In gel electrophoresis, the molecules to be separated are pushed by an electrical field through a gel that contains small pores. The molecules travel through the pores in the gel at a speed that is inversely related to their lengths. This means that a small DNA molecules will travel a greater distance through the gel than will a large DNA molecule. And gel electrophoresis involves an electric field. In particular, this field is applied such that one end of the gel has a positive charge and the other end has a negative charge. Because DNA and RNA are negatively charged molecules, they will be pulled towards the positively charged end of the gel. Proteins, however, are not negatively charged. Thus, when researchers want to separate proteins using gel electrophoresis, they must first mix the proteins with a detergent called sodium dodiosyl sulfate. This treatment makes the protein unfold into a linear shape and coats them with a negatively charge, which allows them to migrate towards the positive end of the gel and be separated finally after the DNA, RNA, or protein molecules have been separated using gel electrophoresis. Bands representing the molecules of different size can be detected. And applications: DNA can be separated by electrophoresis. To visualize bands of a molecular marker to genotype individual plants, and to verify amplification by PCR or sequencing reactions, and to check the quality and quantity of genomic DNA after DNA extraction, to separate DNA fragments to close a specific band. And next application is gel electrophoresis is used to provide genetic information in a wide range of data field. Human DNA can be analyzed to provide evidence in criminal cases, to diagnose genetic diseases, and to solve paternity cases. And the next instrument is blotting technique, southern blotting, northern blotting. Blotting is a technique for detecting DNA, RNA, or protein initially present in a complex mixture. In this technique, the molecules separated by gel electrophoresis as transformed to nitrocellulose filter. Three types of blotting techniques are commonly used for visualizing particular macromolecules. First one, southern blotting. This is a DNA blot transfer. It combines the use of restriction enzyme, electrophoresis, and DNA probes. DNA isolated from cell line or tissue is digested with one or polyacrylamide more restriction enzymes. This mixture is pipetted into a well in an agarose or polyacrylamide gel and electrophoresed. DNA being negatively charged migrates towards the anode. The small fragments move rapidly. After this, the DNA is denatured by exposure to mild alkali and transferred to nitrocellulose or nylon paper, resulting in an exact replica of the pattern on the gel. The DNA is bound to the paper by exposure to heat or UV radiation, and the paper is then exposed to the labeled cDNA probe, which hybridizes the complementary fragment on the filter. After through washing, the paper is exposed to X-ray film, which produce specific bands corresponding to DNA fragment that recognize the sequence in the cDNA probe. And next one is northern blot. This is similar to southern blot. RNA is subjected to electrophoresis before blot transfer. And third one, western blot. This is for protein. Proteins are electrophoresed and transferred to special paper that avidly binds macromolecule and then probed with specific antibody or other probe molecules. And next one, applications of blotting and hybridization technique. Southern blotting techniques is widely used to find specific nucleic acid sequence present in different animals including man. For example, if we want to know whether there is a gene like insulin in sea anemone, then DNA of sea anemone is mobilized or membrane or blotted by using insulin probe against it. And another unblotting technique is widely used to find gene expression and regulation of specific genes. For example, if we find human insulin like gene or oyster, then by isolating and immobilizing RNA and blotting it with insulin probe, we tell whether the gene is expressing or not. By using blotting techniques, we can identify infection infectious agent present in the sample. We can identify the inherited disease, and blotting technique can be applied to mapping restriction sites in single copy gene. Thank you.